Rhodopsin misfolding causes progressive retinal degeneration called retinitis pigmentosa. While a traditional imaging method have limited capacity to quantify the rhodopsin transport, this newly developed image-based assay allows for high-throughput screening, eliminating false positives.
This technique allows for a quick evaluation of drug pharmacodynamics aimed at rescuing the folding of disease-causing rhodopsin, thus accelerating the potential for a treatment for the quietly untreatable blinding disease, retinitis pigmentosa. The cellular localization of a membrane protein is very important for its function. This method can be easily modified and applied to quantify the transport of any other membrane protein of interest.
Make sure you prepare a plate map and gently add an aspirate liquid from each well of the plate to keep cell number and immunostain conditions consistent between the cells. This will yield a high Z factor and reliable results.
To begin, seed 5,000 cells per well into a black wall clear bottom polylysine treated 384 well plate. Incubate the plate at 37 degrees Celsius with 5% CO2 for 3 hours for the cells to attach to the bottom of the plate. Next, use a multi-channel pipette to add 10 microliters per well of 5x working solutions according to a predetermined plate map like the one shown here. Now, add 10 microliters per well of 2% DMSO to columns 22 and 24.